So we're just going to quickly cover the different components and functions of your blood. Your blood is going to maybe technically or just generally lumped in with the circulatory system. We're going to view it as a distinct tissue. And I know it's weird to think of blood as a tissue, but realize it's a group of cells that perform specific functions. So we could classify it as a tissue level type of organization. When we look at your blood, you have 5 key functions that are occurring with the liquid portion of your blood. First up is to transport dissolved substances. When we say dissolved substances, we're talking about things like sugars, gases, waste products, nutrients, hormones, all kinds of different substances that need to be transported around your body. It's tasked with regulating pH and ion levels in your tissues. That's especially key for enzyme function in different regions of your body. So very important for that. It restricts the loss of fluids at sites of injury. So we're going to talk about platelets and how platelets are preventing further bleeding. Defense against toxins and pathogens. We're going to get a lot more detailed about this in an upcoming set of notes, but really looking at your immune system and how is your blood involved, at least partly, in actually doing most of the work or the tasks of identifying pathogens and removing them or eliminating them as a threat, and then distributing heat and stabilizing body temperature. Moving heat from the core to the extremities, and helping with thermoregulation in that region. So plasma is the fluid portion of your blood. If we look at this test tube over here, we've got whole blood would be all of that mixed together directly as it would come out of a blood vessel. When we're talking about plasma, we're talking about what kind of looks sort of yellowy liquid here. This is blood that's been centrifuged or spun at a high speed to separate out the different components. And so when you centrifuge blood, the plasma rises basically to the top because it's less dense, while the solid red blood cells go towards the bottom of the tube. When we're looking at plasma, it's mostly water, about 92% water, with a small portion of other substances. Plasma is the fluid that's responsible for transporting blood cells and platelets, proteins, hormones, ions, dissolved nutrients, and the gases. It's the liquid portion of your blood. And so really when you think of red blood cells, these are actual solid cells, right? And plasma is the medium that they're floating in. Okay, so plasma is the fluid that the red blood cells float in. If you had only red blood cells in your blood, it would basically be solid or semi-solid and would not flow well. When we look at the cellular portion of your blood, or the non-plasma portion that's down here, we have 3 main types of cells or pieces of cells present here. Red blood cells are the cell types that transport oxygen. The oxygen binds to a special protein found in the red blood cell called hemoglobin, which binds oxygen and transports it through your blood. You guys did a whole lab earlier in the year where you looked at some different blood types and the red blood cells of different organisms. And remember, red blood cells in humans lack a nucleus. I don't know if you remember, you looked at like fish and bird red blood cells had a nucleus, and human red blood cells lack a nucleus. They're denucleated cell types. Our white blood cells are a key part of the immune system. We're going to get much more detailed on that later. And then platelets are cell fragments that are involved in the clotting process. Clotting is where you have damage to a blood vessel, maybe a direct cut, tear, abrasion, or even like a rupture, like you might get in a bruise. And these platelets are fragments of cells and proteins that bridge the gap and prevent future fluid loss. Your different blood cell types, red blood cells and white blood cells, are mostly formed in the bone marrow. This is the stem cell type. All right, bone marrow is actually a stem cell, undifferentiated cell types, capable of making red and white blood cells found in the sort of middle, in the porous spaces of our the bones in our arms, legs, and hips. And so you've got these undifferentiated cells that differentiate into the different blood cell types. Remember, differentiate is when you take a stem cell and turn it into maybe an RBC, red blood cell, or a WBC, a white blood cell. And there's different types of white blood cells we're gonna talk about later. But they're gonna mature and then be released out in the body. In a healthy human, we're talking about 4 to 6 million red blood cells per milliliter. Remember, you could have 6 liters or 6,000 milliliters of blood in your body. So if you want to multiply that, you're going to get to some pretty big numbers of red blood cells pretty quickly. The average healthy red blood cell in a non-diseased individual is going to live for about 120 days. So you can think of it as that it's about 4 months, okay? And then they're going to be recycled. Now, they're obviously staggered, so you're not recycling all of your red blood cells at any one point in time, but staggered, and they're being removed constantly and replaced constantly on a daily basis. When we talk about the platelet action and how clotting works, okay, that's sort of what's being pictured here. We've got these would be individual platelets, which when we have a rupture or damage to a blood vessel, what these platelets do is they actually can lock on and bind right to the edges of the cut. And so you get one platelet, and it will build these bridges and connect with protein fibers to other platelets, and sometimes even lock red blood cells in place. And so they basically kind of make like a web. It's like shooting a spider web over the hole in the capillary. And they'll link together and eventually prevent other blood loss from the area, and they eventually solidify, and if this is on your skin, they form what would look like as a scab, what you guys call a scab. And that would be a permanent, then what's called a clot that is able to block blood loss. And then what the platelets allow for is the cells then on either side of the tear to start dividing by mitosis and fill in the damaged region of the blood vessel. And once that happens, these platelets will disintegrate and they'll break away and be filtered out, typically by the kidneys. And so these are a fluid loss preventing portion of your blood. People with clotting disorders like hemophilia, they don't make certain platelet factors, and so they have trouble repairing damaged blood vessels. We like to talk about and frequently refer to blood types, okay? In humans, remember, we've got some different alleles for blood. We can have an A allele, B or O alleles, and you can have different combinations of A, B, and O. You can have AA, AO, BB, BO, AB, or OO allele combinations, which make a variety of different blood types. These are coming, remember, from your parents. So you've got 2 copies of each allele, one from each of the homologous chromosomes provided by your parents. And so there's a genetic determination of blood type. In order to test for this, remember, we did this testing in our genetic lab. We simulated with simulated blood typing by looking for antibody binding, and this is using an antibody-antigen binding type. And so each blood cell on the surface has these little flags, I like to call them, and they identify the cell type. And your blood type is determined by which types of little flags or antigens that you have on your cells. And each blood type has a specific combination. And if you're A, you've got little A flags. And if you're a blood type B, you've got little B flags. And if you're AB, you've got A flags and B flags. And so your body, if you get blood from somebody else, can identify whether that's the proper blood type by looking at the flags. And if you have somebody else's flags, so if you're type B blood and there's blood cells floating around that have little A flags on them, your body will identify that and mount an immune attack. And your body does that by releasing what are called antibodies. Okay, these are a special type of white blood cell that are floating around in your blood all the time, and they're looking to identify and destroy foreign antigens. Antigens, little flags. Remember, the antigens are the flags. Little flags that don't belong there. And so if you have type A blood, okay, you have anti-B antibodies, or antibodies that would attack B-type blood. And so they float around in your body all, and they look for B-type blood. So what would happen if you had blood of the wrong type introduced, maybe a bad transfusion? What we see is that, let's pretend that we're going to put a star here. You're a person with type A antigens, the little flags. Okay, so your little flags have an A on them, and you're making antibodies here that are anti-B, so they're going to attack B-type blood. And so if you look here, these antibodies are actually shaped that they'll attack the little flags present on B-type blood cells. Okay, the thing that's neat is over here, type O. Type O, as you can see, has no antigens. So no little identifying flags, which means you can give type O to anybody because your body can attack them. They can't see that there's any little antigens there. But the consequence of being type O is you attack all other blood types. Okay, so you couldn't receive a transfusion from anybody who's not type O blood. And so what happens is your body reacts to the cells by clumping them together and forming a giant clot, and then it will break them down, all right, into their cellular subcomponents, break them up into little just protein fragments and filter them out with the kidneys. But the danger is that these clots could lodge somewhere like the brain or the heart or a lung and cause an embolism or a stroke or a heart attack, etc. And what that looks like under a microscope is shown on the next slide. And so here you would see this would be an attack. Okay, so this would be normal blood cells, right? And you guys have all seen these. But if you had, let's say you had B antigen here, so cells with little B flags on them, and you also had B antibodies or anti-B antibodies. All right, they would attack the type B cells and make a big clump out of them. And this chart sort of tries to show you here. These are blood combinations, so B with A, that would lead to clumping. Okay, or and if you look, recipients. Okay, if you're O, you can't receive blood from anybody, A, B, or AB. You can only receive blood from O. If you're A, you can receive blood from O and from A and so on and so forth. So this is what that would look like, that clumping down here at the cellular level. This would be, like I said, normal blood cells up top. The extra sort of factor in blood typing is that not only do we have A, B, and O cell types, but we also have what's called an RH factor, sometimes called the D antigen, and this is what determines positive or negative, okay? And so you could have a cell, let's go back and pretend that this is an A, so it's got an antigen on there for A, and it could have a whole nother antigen for RH, and that would make it positive. Okay, individuals that are negative, okay, lack the antigen, but they can attack. They have an antibody that would attack other positive people. Okay, And so B positive blood can't be given to a B negative person, okay, because their negative person would attack the positive blood, but B positive person could take B negative blood. So it's an extra factor that has to be matched up for blood donation purposes. So I always think it's neat to kind of just look. Blood type varies widely by genetic ancestry. So we have certain groups that are 100% Rh positive because of their founder origins. We also have some groups like native South Americans who are 100% O-type blood. Just think, a couple thousand years ago, the people that moved down to South America, everybody that migrated down there had type O blood. Nobody ever mutated and developed any other blood types, and so they still have that percentage, that 100%. Whereas other populations that probably had a lot of mixing have sort of equal amounts A, B, and O type blood. The U.S. average, B is kind of going to be the rarest, pure, and then AB very rare, but most people in the U.S. are going to be of the positive type. So you can look at that chart on your own for something that you find to be interesting. The only disease that we're going to talk about for blood, and there are many, HIV, etc., but we're just going to look at anemia, and anemia is just a general term, very general category or descriptor when a body does not have enough red blood cells for some reason. This could be due to low blood count. Maybe you had a traumatic injury, right? Lost a lot of blood, or recently donated blood. You could have abnormal hemoglobin. You could have sickle cell anemia, which we've talked about in previous units. You could be iron deficient, okay? So you don't have enough iron in your body or in your diet, so that you have trouble making hemoglobin, which is based around an iron atom. All these could lead to low blood cell counts, which leads to things like headache and tiredness, and if it's sickle cell anemia, joint pain could possibly lead to death if your body for some reason isn't making blood cells to replace ones that are lost naturally. And that just the general term there is anemia for those types of diseases.